As Dan mentioned, the research focus in our lab is on C4 photosynthesis. And I was going to tell you about some of the comparative transcriptomics work that we've been doing, which is actually partially funded through an additional CSP grant that we have. That's been really informative, and I'll allude to what we're getting out of that, but it's still early days. So what I decided to do today is tell you about some of our efforts that have been ongoing with JGI to develop resources, genetic resources, informatics tools for Setaria viridis, which is a small panacoid grass. So your first question may be, why do we need another plant model system? It turns out that the panacoid grasses, shown here, are our major bioenergy feedstocks. So in the US, it's pretty much all corn-based ethanol. In Brazil, it's sugar derived from sugarcane. And sorghum is a very interesting player in this space now, where there's potential use both as a sugar-producing crop and also in a grain feedstock, being able to slot into both US and South American bioenergy production. But panacoid grasses are also some of our most promising second-generation lignocellulosic feedstocks, so miscanthus and switchgrass. So this is a clade of grasses that diverge dicots about the panacoid from the poids, like wheat and bracky and rice, about 60 million years ago. And so to us, it actually just made sense that if the target is to engineer these panacoids, then we should be looking for a evolutionarily related model species to facilitate that translation of discovery, basic discovery in your model, into application in your feedstock. And I've listed down here a few of the characters that many of these panacoids share. The one on the bottom there is the one that we're most interested in, C4 photosynthesis. So as you may be familiar, so most plants utilize C3 photosynthesis. It's a single cell type way of fixing carbon. Everything's contained within a green cell. In C4, which is a more efficient form of photosynthesis under hot, dry conditions, that photosynthetic process is actually partitioned between two cell types. So you have CO2 initially fixed in the mesophyll. It's then shuttled over as a C4 acid into the bundle sheath, and it's decarboxylated. And so that two-cell system results in the differential expression of about 65% of the genes that are on in the leaf. So there's been an extensive rewiring of the photosynthetic process. So if we want to engineer that process, again, it makes sense to use a system where it's already split photosynthesis into two cells. We can study that process, identify the regulators, and then potentially engineer improved or gains in photosynthesis in our target feedstocks. So that was largely the rationale. And it's been something I've been thinking about for many, many years, but really we didn't have a good opportunity to move into Setaria because there were a few big bottlenecks. So I've shown here some of the positive attributes of Setaria. So it's a small plant. So it's the smallest of the panacoid grasses. It will flower at 10 centimeters in height. So you can grow this much like you would grow a Arabidopsis. It's got a rapid generation time. So it's 6 weeks from seed to seed if you grow it under short day conditions. One of the bottlenecks that kept us out was that we didn't have a genome sequence for Setaria. And so Jeff Bennetson led an initiative with JGI to sequence the cultivated form of Setaria, which is Setaria italica, or foxtail millet. Foxtail millet is cross-compatible with Setaria viridis. You can get fertile F1 seeds. You can create F2 populations. And in fact, we're using that as a way of exploring the domestication process now. But foxtail millet is still a crop. It's a big plant. It has a long generation time. And so, but the genome sequence is very similar between italica and viridis. So much of the mapping that we do for the expression profiling can be layered on to the Italica genome. And we're now working with JGI, again, to construct a Viridis genome sequence. And so the sequencing is no longer limiting for us. What was limiting was genetic transformation. And so we really started in earnest about 5 years ago, once we knew that the genome was coming, to start to develop the tools for transformation. And Joyce Van Echt at the Boyce Thompson Institute at Cornell University really had the breakthrough and was able to transform Soteria at efficiencies of up to 10%. Joyce has continued to develop that method and improve on that protocol, and I'll show you some evidence in my last slide that suggests that she's now up to about 25% transformation. So transformation now is no longer a bottleneck. We can introduce genes. We're starting to develop constructs using the CRISPR-Cas9 system that we heard about yesterday to do precise engineering within Soteria. And as I mentioned, it's a C4 photosynthetic system. And so this is just a phylogenetic tree that shows you where Soteria sits. So it's a panacoid. This is the panacoid clade within the grasses. And the color coding here represents independent acquisitions of C4 photosynthesis over evolutionary time. And this is one of the really fascinating aspects of C4, is that it was independently co-opted in over 70 lineages of plants from existing 3C systems. So you can see here with the color, it's blue, so it's a C4 NADP malic enzyme subtype. This basically just is a way of categorizing C4 by the decarboxylase. So this is the enzyme that does the primary decarboxylation of the C4 acid. And so you can see that in this lineage here, it acquired this NADP subtype independently of the lineage that has maize, sugarcane, and miscanthus. So what this allows us to do is to now compare and contrast different C4 systems. So we can identify commonalities in terms of transcription factors or potential cis regulatory elements that are shared between these two groups, but we can also look at for points of divergence. And when we see some of those divergence points, again, it provides us opportunities for engineering. So for instance, we see that there are transporters of some of these C4 acids that are present in Soteria that have not been co-opted in maize or in sugarcane and vice versa. So now these are going to be tools for allowing us to do cross-species engineering. And this, I think, is a really exciting place now in plant science. Many of these genomes are currently either being sequenced by JGI or other groups, and again, it will provide us this foundation for moving across these closely related lineages to discover new genes and potentially new mechanisms involved in C4 photosynthesis. Okay, but what I really wanted to focus on today are the points that are highlighted in red here. And these have all been projects that have been facilitated or accelerated by the sequencing that's ongoing at JGI. And so our CSP was to develop community tools for Soteria viridis as a genetic model system. So I mentioned when we started this, we had the genome sequence, we had developed transformation, but we were still limited by performing crosses. So I'll tell you a little bit about what we've done to make that process a little easier. We're creating mutagenized populations now for both forward and reverse genetic screens. And we're starting to characterize natural variation in populations of Soteria, where we're assembling and building a large diversity panel for genome-wide association studies. I think Jerry, on the first day of this meeting, gave you a really good example of the power of GWAS and in Poplar. It's pretty amazing what they've been able to do with fine mapping genetic variation that can basically to fine map any trait that you can phenotype. We'd like to emulate that for Soteria. We're also developing RILs that'll give us a little bit more power to interrogate specific alleles in 2 different parents. And as Dan mentioned also, the Gene Atlas work, where we're trying to profile expression in a number of different tissues. So you can see that the sequencing is touching a number of different aspects of our work. One area where it hasn't touched is on performing controlled crosses. So when we started this, we were told everything from Soteria is actually very difficult to cross to Soteria is impossible to cross, and you've got to go find these magic people in China who know how to do crosses in Soteria. And it's true. Doing crosses in Soteria is not like doing crosses in corn. It's a lot more work, but it is possible. Part of the challenge is that you have small flowers. They're densely packed, so this is a panicle that, on the right here that shows you how closely packed those flowers are. There's also a relatively short time period between flower opening and crossing, and this is actually shown as a time series down here at the bottom. So this is a flower that was just starting to open. You can see here the stigma exerts, and then your anthers push past the stigmatic surface here. The anthers open, and then they dump the pollen, and the flower clamps shut. And so this whole process takes about 40 minutes. So it's tough because if you don't know when this happens, you're going to miss it, right? And you don't want to have to go in and start to rip open the flowers to get out those parts because it's very destructive. So what we did, or more precisely, what Wei did, I'll be talking about a lot of stuff today that Wei was really the driver of. And she, from the moment she stepped into my lab, she's really had a passion to develop Soteria as a model system. And so what Wei did was worked on developing this crossing procedure. She takes a panicle, she trims it to get rid of either flowers that have already been pollinated or flowers that are fairly immature. She does this at a time when most of the flowers look like they're ready to open. So she trims it back. This is probably the most labor-intensive part of that process. This can take about 15 minutes to trim back the flowers. She then gives it a heat treatment, and the heat treatment is just dipping those panicles at 48 degrees for 5 minutes, and that'll kill the pollen. On the second day, so then she puts them back in the chamber. On the second day, what we do is grow these in a well-defined growth condition. So we have a morning cold treatment, which synchronizes that flowering process, so everything opens up at the same time of day, so she can come in at 8:30, have her flowers open. She then takes another panicle as the male performs the crosses, and then 2 weeks later can harvest the seed. So Wei has published a protocol. You can see it at the bottom here. We use the JOVE, the Journal of Visualized Experiments, because it's a very manual process. And so it's a video of her doing the crosses and kind of walking through the process. So if you're interested, I would highly recommend you check out that video. So now everybody in the lab is doing crosses this way. It's similar to Arabidopsis style crosses. So you're doing 10 or so crosses a day. You're not doing hundreds of crosses like you would do with maize. So once you make that F1, though, then the biology can take over. These things are good selfers and very prolific seed producers. So once you make that F1, in the F2, you can harvest hundreds or thousands of seed off of an individual plant. Okay, so where does the sequencing come in? Well, another cornerstone to genetics is mutagenesis, and what we've done now is embarked on a large mutagenesis experiment where we've mutagenized 20,000 M0 seeds to create 20,000 M1 plants. And you can see a snapshot of those plants growing in the greenhouse. We used NMU, N-nitrosomethylurea, as a mutagen. We tried using EMS, and for the life of me, we couldn't get it to work, and I still have no good reason why. We've done very similar things with Brachypodium, That worked fine with EMS. So if anyone has a good idea why EMS wouldn't work and NMU would, I'd be happy to hear it. So regardless, we went ahead, did the NMU. It's an alkylating agent, as I mentioned, so it creates G to A transitions. And one thing we like to see in these M1 plants is evidence that the mutagen is working. And so what we look for are sectors on the plants, and you can see a nice sector here on the leaf. So this would be a dominant mutation that resulted in a most likely a gain of function mutation. And you can see that in this sector that it actually moves up into the panicle. So I don't know if you can see it that well, but these flowers here are pale yellow like this sector on the leaf. And what this does, it really highlights that mosaicism that you see in this M1 generation. So when we build these populations for reverse or forward genetics, we can't work on the M1 populations because of this mosaicism. So what we do is we harvest the seed from these M1s, these M1 plants. That gives us the M2 seed. We then plant out that generation, and that's where we see the segregation of mutant phenotypes. But again, because of that mosaicism in the panicle, we don't expect to see nice 3-to-1 segregation in these M2 families. So what we do is we harvest DNA from a single plant. That'll capture a percentage of all those mutants that we generated in the M1. The DNA is then arrayed in a 96-well plate, and we harvest the seed from that same individual. And then we have a 1-to-1 correspondence of the plant that carries that particular mutation in the seed stock that was derived from that plant. And now those M3 seeds should be showing nice 3-to-1 segregation. So because we plant out all of these families, we actually don't just throw away the seed. We save any any seed from the mutants that we identified, and we save a pool of the other lines. So we've done this on a smaller scale. We piloted about 2,000 families in this way, and that's what enabled us to develop a lot of the resources and protocols that I'll be talking about in the next slide. So when we characterized those M2 populations in that pilot study, again, there's about 2,000 families that we looked at. We saw a nice diversity of phenotypes. There were about 30% of the families that had visible mutant phenotypes, and this is just showing you a few of those phenotypes. And what you would typically do to determine the mutation frequency is look at the frequency of pale greens or yellows and then try to extrapolate back to mutation frequencies. But now in the age of genome sequencing, we can actually empirically determine these mutation frequencies. So what we did is we just selected a number of mutants, identified actually 55, and 27 of those have now been sequenced at JGI. And this just shows a summary of those sequencing efforts. So what's shown across the top is the gene density along the 9 chromosomes of Soteria. So you can see the gene-rich regions are near the ends of the chromosomes, as is common in many plant systems. And then what you see in panel B is just the mutation density along those chromosomes. And so you can see it's a pretty uniform spread across the chromosomes, perhaps a little higher near the centromeres as you might expect, because you can handle a greater mutational load where you don't have as many genes. And then the important graphic shown here is in panel D, and this is a summary of the homozygous disruptive SNPs. So these are mutations that are likely to result in amino acid substitutions, missense, or splice junction mutations. And you can see that the number of these disruptive SNPs is about 50 for the population. And so based on this, we could then extrapolate to what we need in terms of a population size to have a high likelihood of obtaining X number of alleles in any particular gene. And so what we realized here is with about 6,000 plants, about 6,000 M2 lines, we could generate on average about 10 alleles, mutant alleles of any given gene in the genome. So that's really where we're headed now in terms of creating the reverse genetics. In terms of the forward genetics, we're using bulk segregant analysis here. This is just showing you a schematic of how bulk segregant analysis works. What you do is you mutagenize your population. It creates a lot of SNPs, many of which are not linked to your mutation of interest. You then back cross it to the parental line and then self-pollinate that to create a segregating population. You then pool the individual mutants and sequence that deep pool. So there'll be a number of SNPs that are present in that mutant population. You can map those back to the genome. But what you're looking for are those SNPs that are fixed in the mutant alleles and not segregating. And so what you've done is shown linkage in this way with the SNP with your particular mutant phenotype. So this technique has been used very effectively in Arabidopsis and rice to create and find map many mutations. So we're setting up this procedure in the lab now. We've decided to target one of our sparse panicle mutants as a proof of concept. And we've made it down to this point where we're now we've made the DNA, or we'll set up to deep sequence. And so what's important to look at here in the left is the timeline. So we can move from discovery of a phenotype to the actual gene in hand in as little as 7 months with Soteria. We're also setting up a reverse genetic screen using tilling. This is a procedure where it's a PCR-based screen to identify mutations in the gene of interest. And traditionally, the way this has been performed is with a relatively labor-intensive detection method that involves forming duplex DNA. If there's a polymorphism in your PCR amplified region and wild type, it creates a bubble. Cell 1 is an enzyme that'll cleave that heteroduplex, and then you have to visualize it on a gel. So because of these labor-intensive steps, it makes it a relatively costly and time-consuming process. So what we're doing now is, again, going back to sequencing, using sequence analysis instead of gel-based detection systems to amplify and then sequence deeply across these pools. So we create these super pools where each well has 12 individuals, and then we pool across. So we'd have 144 individuals for PCR amplification. And then by indexing these PCR products with primers, we can then deconvolute and end up identifying an intersection where we have a SNP that's present in a row pool and a column pool, and that should tell us which one of 12 plants are we hitting. So we developed this as a proof of concept with Brachipodium. We were able to do sequence tilling by sequence to identify mutations in about 6 genes to date, and on average we were getting 3 to 4 alleles for each gene. So we don't anticipate too many problems ramping it up now for Soteria. We're also using sequencing to characterize a diverse population. So to date, we've been able to assemble a population of about 430 accessions from our colleagues. And we're now sequencing many of those accessions here at JGI. Pu Huang has started to look at the population structure in these lines, and you can see 3 strong subpopulations from these materials. And this is of the 187 lines that have already been sequenced. So, I think this is going to provide us with an excellent foundation for doing GWAS. You can see here that those subpopulations are actually tracking to geography. And if you're real good, you can see that many of these collections follow the major highways across the U.S. So we're at that point now where we're not limited by sequence, we're limited by seed. And so Pu has been going on collecting trips around the country. So we send him into far out regions to go pick up Soteria at the side of the road. We're also creating real populations. Again, here we're using sequencing to find map breakpoints. So we create a cross between 2 diverse lines and then use GBS to define the breakpoints in the population. So Again, what we're limited by now is the time that it takes to construct these populations, not any sort of analysis of where the breakpoints are in the population. The Gene Atlas is currently ongoing as well. Here we've used both A10, which is the weedy, and B100, which is the domesticated, so we can begin to look at potential targets of selection in terms of gene expression. I think this will be a really useful resource. And finally, this is some of the work that Joyce Van Echt has really pushed on, and this is developing not a new protocol, but actually just using a different accession of Soteria to get a much higher transformation efficiency. And when you look at that, the process of transformation broadly across plants, usually it's not a breakthrough in the protocol, it's a breakthrough in finding the right genotype that facilitates the transformation process. And so as part of a collaboration with a number of groups, Danforth included, we surveyed about 60 different accessions of Soteria, looking for those that produce the best callus. And this line here, ME34V, so far has the highest rate of transformation. So seed stocks have been submitted to USDA, so you can obtain seed there. So what I hope I've done today is sort of given you a highlight and giving you an idea of how we can use sequence analysis to greatly accelerate tool development in this model system. And I think we're at an excellent point now. We're really poised to exploit a lot of these resources in Soteria and do fundamental discovery in this panacoid grass. So with that, I'd just like to thank the folks in the lab who've been driving this, particular Wei Zhang, who's done all of the mutagenesis, set up all of the mutagenesis I described, as well as developing the crossing protocol, and has been distributing seed to pretty much anyone who's asked for it. Pu Wang is leading our efforts on the association studies in the lab, and of course the guys at Alpha Hudson and JGI for all of the wonderful sequence that we've been receiving, and we really do appreciate all the help. And with that, I'd be happy to take a question or two.
Thanks, Tom. We have time for a question. Tom, can you comment on the Cas9 CRISPR as a tool that you might be adding?
Yeah, so we're just now getting that into tissue culture and getting some events. We've designed the system to target a couple of genes that we know will give a strong phenotype. I know John Vogel's had good success with it in brachy. Others have demonstrated it in maize and in rice. So it's been proven to work in the grasses. One of the things that we've been struggling with is having one selectable marker. So we need to set everything up on one construct, which isn't too bad. But that's really where we're at. I think it's a very promising technology, though.
Cool. Laura.
I think it's great that you're developing all these resources, and it should be really useful to the community. Have you? It's also great that you're willing to share them, but that's kind of probably not sustainable for a single lab. Have you thought about more sustainable distribution systems?
Yeah, so we've set up an agreement in place with USDA. They're going to distribute all of the lines that were, all of the natural accessions that we're sequencing deeply. We've deposited about 60 accessions now with David Brenner. You can access those. If you look at the website here at the bottom, it's not the easiest web link to get to. But if you go to our homepage, you can find links to these resources. Seed distribution, though, of the mutagenized populations is more challenging. I mean, it takes a lot of time to simply put seed into envelopes. I mean, it doesn't sound like it should, but it does. So if people have wanted to have access to the mutagenized populations, what we've been doing is saying, just send your people to our lab, have them spend 3 days putting seed in envelopes, and then they can take them home with them. It's not a sustainable approach, but right now it's what we've got as a short-term fix.
I guess you can't just plant them along the medians of the highways and then give people a
Yeah,
geographic,
that's-
you want that mutant? Go to Topeka, right?
That's exactly.
Any other questions? If not, thanks again, Tom.
All right, thanks.